What's on the exam?
What's on the exam? You have 2 past exams you can look. So you've got a set of study guides, right? So it's all the stuff on the study guides for the chapters we've covered so far. Yes.
On the study guides, there was chapter 10, but we haven't actually gone over chapter 10.
So we're not doing. So chapter 10 is, I think, signaling, and we'll go over that later. So, but the chapters we've done so far, but then with, there are things that are more important than other things, right? In terms of that. And if you look back at the old tests, those 2 old tests, you should get a feel for kind of the sort of things that'll be on the exam. Also, if something appears twice, appears on both tests, probably was pretty important. Certainly in terms of, I haven't written it yet, but, knowing a step in glycolysis. So if I ask in terms of a pathway, I'm going to ask, here's this step. I might give you the leading up steps or the steps before and after, and say what goes here in the middle. Okay, so I'm going to ask you for a piece of it, not to write the whole pathway. Glycolysis is really important. Certainly gluconeogenesis, the citric acid cycle from that perspective, that you would be able to draw overall metabolism for carbohydrates, right, at this point. So starting with polymers and going down to where you've essentially made ATP. That diagram's important. I'm just thinking about most important things off the top of my head. Certainly give you a problem on adding up ATP. Okay, so how many ATP are produced if you start with this thing? Maybe it's something that isn't part of those processes, but I'd give you a reaction to insert it in, like that malate problem, I think it was. In the case we start with malate. More stuff than that, but electrochemistry problem. Just trying to walk my way back here. Theoretical electron transport chain problem. Regulation of, so we didn't talk, we never had a quiz that did used fructose 2,6 bisphosphate in terms of how it regulates things, but it is pretty important. So that's something that you might see, and then certain things you've seen on quizzes are important. You might see those again. Yes.
I have a question on the exam one.
Mm-hmm.
It talks about, like, we talked about how AMP is really signal, so you don't get a lot of it.
Mm-hmm.
And then it talks about like high levels of AMP.
Yeah.
And then spontaneous reactions. How are we supposed to know which enzymes are affected by it?
Let's look at that particular problem.
Question like B and C.
You said 2023?
Yeah.
Okay. Let's grab the non-key version. There. Okay, so which problem are we looking at?
First page, B and C. Question 1 and C.
Here?
2.
Am I on the right?
Second page.
Oh, but what is B?
Oh, sorry, one more page down.
Okay.
One page down. B and C. Page 2. Yeah.
Here. Okay. So this is a question. Glycolysis and gluconeogenesis do not run at the same time, okay? When enzymes are in one pathway are activated, the corresponding enzymes in the other pathway are inactivated. All right, and so the question is B and C. How will enzymes in glycolysis and gluconeogenesis be affected by high levels of cyclic AMP? So address them separately. So meaning I want you to tell me what happens to each, not just tell me one when I say stuff like that. So cyclic AMP, what is that signal? So there are 2 types of signaling questions we would get in this class. There'd be you've memorized that signal molecule. Fructose 2,6 bisphosphate is a signal molecule. It does not indicate anything about energy state on its own. Does that make sense? It doesn't get used in metabolism. So it's not a byproduct of something that tells you, hey, the cell is low on energy. It's used as a result of kind of telling you what's going on outside the cell, right? because it is made or torn down based on the presence of insulin or glucagon in blood. Does that make sense? So that is a straight out signaling you memorized what it does molecule. The other one is you can rationalize what the energy state of the cell is based on what is present. And so if we think about, so B is how's AMP going to glycolysis? Let me jump back over to these notes real quick. So we've got,
So like AMP is a potent signal to make ATP.
yes. So glycolysis is going to take us down to pyruvate, right? I'm not going to do the whole thing. I'm just going to say ATP, okay? This would not get you much credit as the overall reaction, all right? So, but when we run glycolysis, we're going to make more ATP. I should do this as glucose, right? And glycolysis here. And then we could talk about gluconeogenesis coming this way. All right, so if we have ATP, ADP, or AMP around, the way we get it is that ATP starts being used, let's say, by your muscles. We get ADP and phosphate made. We can then have two ADP can come together to make an AMP and an ATP. So the phosphate from one of those ADPs goes to one of the other ADPs to make an AMP and an ATP, or to make an ATP. Does that make sense? What's happened there? Okay. And so now this shows that I'm using a lot of ATP if I'm got AMP at a high concentration. And if I'm using a lot of it and it shows that ATP is low, that means that I need to make more, right? So AMP, if we thought about these processes here, AMP is going to take glycolysis and it's going to upregulate it. And it's going to take gluconeogenesis. If you need more ATP, you're not going to use ATP to make glucose. And so you would inhibit that. Does that help clear it up?
That makes sense, but like, how are we supposed to know which enzymes it regulates specifically?
Oh, well, so what enzymes are good for regulation? Yeah, phosphofructokinase 1 and, yep, you're supposed to have these guys memorized, right? Let's go back to this thing. So I've given you the pathway right there, right? So which enzymes are going to be affected by that?
Any that use ATP, ADP?
No.
No. Just phosphate.
That's actually, it's a good guess, right? Any of these, it's which enzymes are good for regulating glycolysis and gluconeogenesis?
Non-reversible ones.
The non-reversible ones, right? So I could regulate here, here, and here because those are unidirectional reactions, right? So for glycolysis, hexokinase, phosphofructokinase 1, and pyruvate kinase. And for gluconeogenesis, pyruvate carboxylase, phosphoenolpyruvate carboxyl kinase, fructose bisphosphatase, and glucose 6-phosphate. Does that make sense? So this problem is testing the B. This is testing whether you understand that those single direction arrows are the ones where you would regulate. Now this is the commitment step. This one's more important, but all of them are sites where you could do it. And then C, which enzymes? Okay, so B is how, right? So glycolysis would be activated. And C is which ones?
Oh, the ones that-
What was that?
the ones that we just talked
It's the ones, it's the single direction reactions, all of them. and name all that apply is your hint there. That don't just give me phosphofructokinase 1 and bisphosphatase. Yes?
So the presence of AMP means that ATP is low?
AMP means ATP is low, correct. ADP also means that ATP is low because you have a pool of stuff that could be either ATP, ADP, or AMP, and it's supposed to pretty much all be ATP all the time, if that makes sense. Your body wants to hold it all at ATP as much as possible. So one way to think about that is, so these molecules could be, ATP can be a signal molecule, but in the end, you don't want, in terms of always keeping your ATP at, let's say, 5 millimolar in cells. The deal there is that it's like your gas tank in your car. You don't want how fast your car to go to be based on how full your gas tank is, right? You want your car to go as fast, full speed until all the gas is gone. If that makes sense or not. You always want to have that ATP up at a level where it's not going to regulate the gas tank. I guess that's a better example for something else that we'll get to later.
We have a question. On
Yeah.
row C, if you scroll down, Barry.
Mm-hmm.
If it says just gluconeogenesis, then are we just naming the ones, the like 3 for gluconeogenesis, not all 6?
4 for gluconeogenesis.
Well, yeah.
Yes, yes.
Okay.
But since it just said gluconeogenesis, yeah, you would do that. Okay, good questions. What else?
Sorry.
Hmm?
For like all the stuff, like I know we went into like photosynthesis stuff.
Hmm?
How much of that needs to be like memorized completely? Like just all of it? I know we kind of did like a lot of.
Yeah, you don't need for the Calvin cycle to know all that stuff, but you do need to know that Z diagram, right?
Look at that over.
Sure.
Oh, but we don't need another Calvin.
What was that?
We don't need another Calvin.
You don't need to know the Calvin cycle in terms of every atom sort of thing going through it. You should generally, though, be able to describe for the Calvin cycle. Let me not write on the board. But for the Calvin cycle, you should be able to generally talk about. There we go. So what the Calvin cycle is generally doing is you have 5. This is the ribulose 1,5-ribulose bisphosphate, right? But we have 3. We have 3 times 5 carbon molecules, right? We add 3 CO2 to that. This is part of Rubisco is going to be the first 2 things here, and that's going to give us 3 times 6 carbon molecules. Okay, so we had 5 carbon molecules. We added 1 carbon dioxide to each, right? Not as carbon dioxide, but carbon. And then those 6 split up into each split in half, and we end up with 6 times 3 carbon molecules. And then the rest of the process here is regenerating those. Okay? So we're going to go ahead and regenerate those to make our 3 5-carbon molecules. And part of that process, we actually pull 1 3-carbon molecule out of it. So for every Calvin cycle, you're going to take 3 CO2 and you're going to turn them into a glyceraldehyde 3-phosphate, right? This is the process. You start with these 5. You add carbon dioxide to make them these 3 things 6 carbons long. You split them in half, you pull 1 off, and you regenerate your 3 carbon, and then you do it again. So that, in general, is what the Calvin cycle does, and you should know that, right? You don't so much need to tell me that Rubisco is important for this, but you need to know things about Rubisco, right? In particular, something that we covered near the end that I want to make sure you think about is we talked about regulation, right? We had a really cool example of how everything that's happening in the light reactions causes conditions to be such that what we need to do, we have a lysine in here. I'll do it unprotonated. So we had a lysine in the active site of rubisco that can react with carbon dioxide to make a carbamate. And this is important in terms of regulation of rubisco, right? This carbamate can coordinate with it coordinates magnesium. So when I say coordinates it, it's holding it in the active site. And that magnesium, I don't know, well actually we figured out what it did. It was actually holding on to a 3-carbon, it was probably a glyceraldehyde 3-5. No, it was the 5-carbon thing from the crystal structure, right? You don't need to know that. But what it was doing in the crystal structure we had was that molecule that it was coordinating to was our ribulose 1,5-bisphosphate. But it needs to coordinate. The important thing is it's like for MTHFS where you have a magnesium that you have aspartates holding on. In this case, this carbonate will hold on to the magnesium. When it's not there, the magnesium leaves. Okay, and rubisco can't run. And if you don't have carbon dioxide, if you have a bunch of carbon dioxide around, you drive this reaction this way, right? And if you have a bunch of carbon dioxide around, you can run the Calvin cycle and make stuff. Does that make sense for that part? And then the other part here is when the light reactions are running, the stroma has a low concentration of acid in it, right? You've pumped all the protons into the thylakoid lumen. And that's also going to Le Chatelier's principle pull it over this way. So you don't want to run the dark reactions if the light reactions aren't running, because you're not making ATP and NADPH to run the dark reactions. If the light reactions aren't running, the pH won't be high. Low protons, high pH. The pH won't be high in the stroma. Okay, so that was late breaking, right, in terms of the stuff we're covering, but it's, I don't know whether I'll ask it or not, depending on how but it's really important. It's a nice rational way that things work. It's an example of evolution taking all the conditions that are going on and having an enzyme that actually reacts in its regulation to that. It's like when we talked about hemoglobin responding to multiple things in a part, like in your tissues, to change to the form that releases oxygen. So really cool stuff, actually. Yeah.
Can we clarify like where these things are? Like is rubisco in the stroma?
Rubisco is in stroma. The dark reactions happen in the stroma. So if I was to attempt to draw a chloroplast, right? The chloroplast has these thylakoid stacks, right? Like that. Okay, and so
So the carbonate is also in the stroma.
the carbamate, yes, that's the carbamate is part of rubisco. So when I draw this line right here is like the backbone of rubisco. This is just one of the side chains. This is rubisco. Good questions. And so what we've got here is if we look at the light reactions, what the light reactions are going to do, let's just call this box the light reactions. We'll call it the light reaction Z diagram. Actually, I could go ahead and draw a Z. Let's see. It's going to go blop, 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 blop. Okay, that's what we get for that. And what that's doing is that's pumping protons this way into the thylakoid lumen. Our stroma's out here. Dark reactions happen out here. Okay. Light reactions happen in the membrane. And then we've got our old friend ATP synthase over here. Okay. With the orientation that I'm showing, those protons can come back out. And when they do, we're going to make some ATP out here in the stroma where we need it. Drawing's a little bit small there. Has everybody got that? Does that make sense?
Can you say one more time, like Rubisco, is like how it's playing in both of them still?
So Rubisco is part of the dark reactions, right? So Rubisco is out here. I'm just going to write Rubisco. The dark reactions are more than Rubisco, right? There's more going on there than Rubisco. Mostly we just focused on Rubisco. Yes.
So we talked about Le Chateliers, like with proton concentration. If there's less hydrogens in the stroma, there's less carbon being made.
Okay. So if there's less hydrogens in here, there's more of making the carbonate. Because Le Chatelier's principle shifts it over, and there's going to be fewer here while the light reactions are going because they are pushing protons in here and giving you this really great gradient. It's like 1,000 times as much H plus in here as out here.
So when we talk about, does the dark reactions and the light reactions, they don't run at the same time?
They do.
They do run at the same time?
Yep.
So if the light
dark reactions and light reactions. Dark reactions do not run unless the light reactions are running.
When we're talking about the light reactions running, like the hydrogens go into the room first, but then they're brought back out to the ATP synthase.
Yes.
So when we're talking about the light reactions running, does that significantly change the concentration, or like does that cause a
that's a good question. So there are 1,000 times the pH in the lumen is 3 units lower than in the stroma when the light reactions are running. 1,000 times as much H plus in the lumen, the thylakoid lumen, as in the stroma when those are running. So yes, you get that effect. So good questions. It is 3 times lower, which means you have 1,000 times as much H plus, because pH is a log scale.
So then when the reactions are running, that decreases the amount of protons that are in the stroma.
Yes.
Within the carbonate. So running light reactions, it like drives more dark reactions.
Yes.
Okay.
Running the light reactions activates Rubisco. Right here, activates Rubisco, which is what you want. The reason why these dark reactions don't happen when it is not light out, when the light reactions aren't happening, is the light reactions are making the ATP. Let me write that. So the light reactions make the ATP and NADPH needed to fuel the dark reactions. Okay, that's really important. These are not storage molecules, right? ATP is an energy currency. There is not much of it in the cell. All right, it gets used and it gets regenerated right away. Same sort of thing with NADPH. These are not, you don't store energy there. You store energy as glycogen or starch or something else. And these are your currencies that you're going to try and hold ATP at 5 millimolar all the time. So when something needs ATP, it's there. And the same sort of thing with the NADPH. But you're not making these when you don't have the light reactions running. So you don't want to use them, right? Yes.
Okay, this is like a really basic question.
Huh?
But if all the H plus is constantly in the stroma or the lumen,
right?
How does the gradient keep going?
How does it keep pushing more in? Well, some's coming out at a rate to make your ATP.
But doesn't a gradient work from most concentrated to least? Like, why would it keep going at a more concentrated?
Because you're using. So the question is, how do you pump these protons uphill?
Yeah.
And you're using the light from the sun for that. You're powering that with the light from the sun. You're using that energy to push them uphill.
But why would it rather do that than just?
Well, you don't because you don't have a hole. These things are these are carriers, not channels, right?
Yeah.
So you are I don't actually know the mechanism of pushing them in, right? We know the mechanism of them coming out,
Yeah.
and we could actually push them in if we dumped a ton of ATP here, right? Let's say you had a chloroplast and you could inject ATP in there. I don't know if people can do that. They probably can, but I don't know if chloroplasts are. Let's say you could inject a bunch of ATP here. It's a reversible enzyme, the ATPase. So you could actually use it to pump protons in. You're just going to run it in reverse. Yes?
Can we look at the whole Z diagram again? Because, like, just to see.
Yes.
Talk about, like, what NADPH does.
Right, right. Before I go look at that diagram, anybody have more questions about these? Okay. So let's look at it. Let's look at it with the old diagram I drew. I can also draw it out. So this is actually the one we looked at. I can also draw it again if it helps to see it drawn. But okay, so here's our diagram. This is posted. So we'll go ahead and erase a little bit of it. Let's erase this. Let's erase this. Okay, so talking about this, you don't need to worry about knowing what photosystem 2 and photosystem 1 is. Okay. But we've got water. All right, we have that electron, right, we have the oxygen evolving complex, really. That is the name. But we pull electrons away from water to make P680 reduced, right? Okay. P680 then is excited. That reduced form of P680 is super stable. Its complement, the oxidized form, is a super powerful oxidant because it's taking electrons away from water. And we know water is really stable, right? Our bodies aren't reacting. I mean, you've got reactions to use water, but water isn't this super reactive thing. We know that. So we pull those electrons away from water. Super hard to do. Now this is super, super stable, and so you hit it with light to make it excited. Okay? And then it can give up an excited electron to plastiquinone. Okay. Plastiquinone will then donate its electrons to cytochrome B6F. Okay, and this is like our complexes in the electron transport chain. Those electrons go across this, and as they go across this, they get lower and lower in energy. And getting lower and lower in energy, some of that energy is captured and used to pump protons across the membrane. Okay, so into the thylakoid lumen. Stroma out here. All right, then those electrons get handed off to plasticyanine. Plasticyanine then hands the electrons off to P700. Once again, we're really kind of low in energy here. Okay, very stable, reduced form P700. We have to hit it with energy equivalent to 700 nanometers of light. That will kick it up and make it excited. It can then give those electrons to ferredoxin, which then hands them off to NADP plus to make NADPH. Okay. And then the ratios that these happen is we're going to get, every time we do that, we're going to get 2 NADPH and we're going to get 3 ATP for that process. And those are the exact ratios we need to run the dark reactions. So a 3-to-2 ratio, ATP to NADPH. Reese, did you have a question?
Yeah, so I know, like, when you're exciting, like, the P680 and the P780, is that going across, like,
the membrane? No.
Yes. Okay.
No. The Z diagram here is just showing, and this Z, so we've got a membrane here, right, to talk about this bit. But the Z is about energy. So higher energy, lower energy, higher energy, lower, 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 higher energy, lower, lower. Okay? And when we think about the energy, we're talking about the energy of those electrons. Right? In many of these cases, electrons come along with hydrogens, but it's about electrons.
And this is the one, I guess we need to know for the things, yeah, not that other one that we talked about first.
The first one, I was hoping that helped because I showed you, so let's take a look at that. So this is the one I would ask. But the first one, these are oxidation reduction reactions, so I thought it was important to show them. Right. So here, where did it show up? This is a better representation because it's showing down here. You donate the electrons, and you make an oxygen from water. You take this from water, you make an oxygen. You go up here, you're taking electrons, plastiquinone grabs electrons, it becomes plastiquinol. It then gives up its electrons, it becomes plastiquinone again. This is like what's going on the electron transport chain, except I'm showing the Z diagram with those, with the oxidation reduction pairs. But you don't need to draw that one. It ends up being a little bit of a mess trying to get it all put together. And that's not how it's drawn in the book textbooks. Yes?
Do we need to know anything specific about cytosol BCZ or plastic cytosol?
You don't need, this is like complex 1.
Yeah.
Right, so you need to know pumps protons as electrons go through.
But there's no like specific thing to know about it.
You do not need to know anything specific about it beyond that. You don't even need to know how many protons. We could work it out. We make what, 3 ATP for 8 electrons going through. You know, so we could work out what our stoichiometry is there. We don't really need to worry about that. I gave you a stoichiometry for these reactions. I don't care that you know that. Okay. That's more just about kind of understanding more about thinking about photons and electrons and how things are going. In terms of plastiquinone, you should be able to recognize it and draw the other form, but I would tell you whether you were in a chloroplast or a mitochondria. Okay, so essentially the plastiquinone and ubiquinone are the same molecule, except that one is in the mitochondria. So here's plastiquinone. So if you want to make that into plastiquinol, how do you do it? What do you do?
Use some ketones.
Yeah, right. So this is going to be, and so these bits are different, right? These are methoxys in ubiquinone. You don't need to know that because I'm going to tell you chloroplast or mitochondria. Okay, and then this tail's a little bit different. Okay, but this is the oxidized form. All right, and so then the reduced form, let me do an arrow. The reduced form is going to look like this. I'm going to go ahead and just throw my bonds in. I'm going to do this, right? Okay, so it's the same thing that you do when you're talking about ubiquinol, ubiquinone and ubiquinol.
This is the chloroplast.
But this is in the chloroplast. Yeah.
Can we go over like all the ATP counting? Like,
Do an example of an ATP counting.
not even that, but just, sorry, like,
It's okay. Everybody's sick right now.
like how it goes, like through glycolysis and like ETC, like how much is produced in each.
Yes.
Yeah.
Would you? Shall we do one more? You don't start with glucose.
Yeah.
Okay.
Yeah.
So I can complicate that up. Does anybody else want to ask anything about the other stuff before we do that?
Was it ferridoxin? Was that the like last thing?
I'm sorry. What was that?
Ferridoxin.
Yeah.
Oh.
What is that? What is that doing? It's going back into that middle.
Does that just decide if the
Oh, for the cyclic electron flow?
Yes.
Yes. Okay. So ferridoxin, it's a protein, it has irons and it has sulfurs in it, and the oxidation states aren't clean. Okay, so it's not. I can say it's an iron +2 or an iron +3. So ferridoxin has the electrons. It can hand those electrons off to NADPH. But if the cell if it's important for that cell to actually have more ATP relative to NADPH, and sometimes it is, and I don't really know what the conditions are, you can take those electrons and pass them back to this complex, and you can just run them in a circle, pumping them around that circle using the energy from this 700 nanometer light, right? So you pump them up, they go back down to here, lower energy, pump them up, high energy, lower energy, except for coming from there, all right? And every time they run through this B6F complex, you're going to go ahead and pump protons across the membrane. So when we think about running this, right, we have 4 electrons that go through. It's actually 4 electrons that go through for 3 ATP, right? There's 4 electrons, 8 photons. So you can imagine every 4 cycles you do with this with an electron, you're going to be able to make 3 ATP for that. Yes. Cyclic electron flow?
No, the four
No.
electrons and the 8 photons.
No. We didn't talk about it, but it's.
Is there still like not a specific situation where you want to go for the cyclic electron flow versus?
I'm not going to ask about that, but if you do it, is water oxidized? If you're running a cyclic electron flow, do you produce oxygen? No, because you don't need to grab electrons from oxygen. Or from water. You're just running the same electrons around in a circle here. Okay. That doesn't mean that the situation is that you don't do it because you don't want to oxidize water, right? Because there's lots of water around kind of for anything. But it's thought photosynthesis started here in organisms that just do this. Right? They get some electrons from somewhere, and then they just run them in circles as opposed to making oxygen. Okay, so the question of doing ATP counting problem. Let's go ahead and grab a couple pathways. I'm not going to ask you to do ATP counting without pathways to look at. Okay, so to think about it, keep in mind that this pathway I'm grabbing right now has, it doesn't show the double ATPs in the bottom. Does that make sense? So it's, you're going to want to be careful with that. Or you could have one that shows the double ATPs, but I gave you a problem where you start late and you only have one of those molecules. Okay, so you need to understand that sort of thing. And then let's go ahead and grab the citric acid cycle. I expect you to know that the pyruvate dehydrogenase complex produces 1 NADPH, or sorry, 1 NADH per pyruvate turned into acetyl-CoA. So I expect you to know that overall reaction. But otherwise, I would give you the pathways if I was going to have you doing this, or give you enough of the pathways, because if I